Yeah, it is my huge pleasure to introduce Professor Ji. He is a member of the Chinese Academy of Sciences, professor and director of the Institute of Primate Translational Medicine at Kunming University of Science and Technology, as well as Yunnan Key Laboratory of Primate Biomedical Research. And he's currently the president of Chinese Society for Stem Cell Research. Professor Ji has been engaged in primate reproductive and developmental biology for a long time, more than 3 decades. And he has made many significant discoveries in this field. His lab reported the first gene-modified rhesus and cynomolgus monkeys via Cas9 RNA-mediated gene targeting in 2014. And they also proved the feasibility of generating chimeric monkeys using embryonic stem cells in 2015, and succeeded in the extended in vitro culture for both monkey and human early embryos in 2019. So he's an extremely prolific researcher, and I'll pass the time over to Professor Ji.
Thank you, Dr. Liu, give a very kindly introduction. And so I might talk. So today my talk is dissecting post-implantation embryos, development and human and monkeys use a long-time culture in vitro. So, and firstly, you know, the life starts from sperm and oocyte fertilization. After this, we made a lot of problems, including the health problem, the disease problem. But I think the first important thing is we have to understand the post-implantation embryos regulation and what's mechanism about the future development. However, because limited by the technique and ethic, we do not so far understand clearly about the embryo development after implantation. Everyone knows here show you slides is rodent and primate embryo development structure and lineage very different from those two species. And also we have problem I just mentioned that we have a 14 days row is illegal and regulated the limited in vitro human embryos research to period before the gastrulation. And so we need the urgent to establish a new research system to answer the questions such as how to embryos and extract embryonic lineage, include amniotic membranes, sac development, and how to establish embryonic structure such as a primary streak and the germ cells organ and the differentiation and so on. So, and in years 2018, have 2 papers already published the development of the human embryos development in vitro up to 13 days. However, this technique is used to deculture system. This shortage about this 2 papers is about the culture embryos largely flat, not like a 3D like in vivo. And another question is unequivocal in identifying cell types such as the PGC, cavities, and so on. So we need the new methods to resolve these problems. So in my lab, we developed a new system called 3D culture system. The development support human plastics through pro-streak lineage. In this method, we add the metric gel on the fertilization embryos at day 9 And then we go to the in vitro for the development until to 14 days. You can see we got a very nice structure for the embryos. And also we found a lot of the new evidence is not reported before, such as self-organization of 3D archer over embryo development. Embryonic disc happened in day 12, and embryo in the then 12 also primary and primate unique sequence single cell technologies to study a lot of the gene expressions, such as transcription factors and the KGG pathway and so on. And also we found the amniotic epithelium and the epiplast, the separation. And we found AME is weakly expressed pluripotent genes. The separation epiplast is related with the asymmetric distribution of EK18 and the firm form-based membranes. So here you also can see and very different from rodent and humans and the different gastrulation embryos. And we also found the anterior vesicle endotherms in these culture systems. And finally, we also found primitive streak analysis have different genes expression profile from epiplast about on day 14. Therefore, also we found that the trophoblast lineage development during these procedures from the implantation day 9 to day 12. And epiplast development, the pluripotent transition, we found some genes very important, such as the DFCP2L1, KF17, and the KF4 and the CD24 is very important for this pluripotent transition. In the summary, we use this system. We found the very clearly embryonic disc, amniotic epiphysis, epiplast separation, basement membrane primary and the primate unique secondary EXAC, and so on. This paper, if you're interested, you can see this here in March. And this work is majorly response by the Professor Tianqin Li from my lab. However, because the 14 days rule limitation, we still have a lot of questions not so understand, such as the molecular mechanism controlling generation of development primitive streak, and the key landmark and the mechanism of gastrulation, endotherm, mesotherm, and the ectotherm lineages specification and the PGC development and so on. So we developed, we use, this time we use monkey embryos with the when the system can culture the monkey embryos from day 7 to until to day 20. So especially very important in day 15, we can find the AM and the VE, AP+, API, and SYS, this you can not clearly find the 14 days before. Also, we use very strong techniques, single cells, to compare the in vivo and the in vitro embryos development different or same. Our work is found this is very same, the in vitro and the in vivo monkey embryos developments during the after the 14 days to 20 days. And this work also demonstrates that culture embryos can form embryonic and embryogenesis, include amniotic membranes EX cell structure, especially the specific specialization of amniotic cells, which has not been previously reported. And also this is amniotic cells express CDX2, OCT4, and TFIP2C. Of course, use the monkey embryos, we can study the crosstalk between cell lineage. The expression pattern of amniotic epithetal cell to that of embryo extra cells, member cells, and spare cells, which are regulated with TGF signals. The cellular connection between trophoblast ectotherm, exotome, and the exotherm interstitial cells and the parametric endotherms. And also we found the winter signals is involved in the formation primitive streak. The winter function for monkey embryos, PS, is formation with psychiatric winters techniques. So we found the chromatin structure is enhanced region of winter cycle pathway membrane are more open in API, EAPI, and the gastrulation cell compared to other cell types. And this also we found the oxidative phosphorylation is very important in the pluripotency regulation. And we also found that the array from the PGC is have two resources. One is the PGC are from amniotic epithelium. And the second one is the PGC are also from EPI cells and similar to early stage PGC in vivo. Actually, this work already published in last year in Science. If you're interested, you can look for these papers on Science. And this work is response by Dr. Tao Tang and Dr. Yu Liu. They're from different group from my lab. And after this, we also compare the human and the monkey embryo development after implantation, the difference or similarly. First, we found the similarly from human and the monkeys is about the formation omnial cavities is from day 8 to 9. And clearly, amnion on day 12 in human and the monkeys. And we also have some found some very different from human and the monkeys, especially for the EPI and the monkeys have a unique phenotype and also some difference. That's especially for Wnt and FGF signal pathway, more activity in human API than monkeys. So this is a general talk about today. I think just to catch time. If you have any questions, please. Thank you.
Thank you so much, Professor Ji. Let's see if there are any questions for Prof. Ji. Perhaps I could get started with a question for Prof. Ji. So I was curious, what do you think is the consequence of more Wnt or FGF or HOX signaling activation in the human cells? So I think on your second last slide.
Excuse me, I cannot clearly hear you.
Oh. Hi, can you hear me now?
Yes.
Yeah. So I was wondering what is a consequence, what's the result of having more activation of the Wnt and FGF or HOX signaling in the human cells?
Yes, and it's a good question. I think for human embryos, the FGF is really important, and but also I think in monkeys.
Mm-hmm. But does it contribute to like the differences that we see between humans and monkeys?
Yeah. Well,
Yeah.
this difference is actually is for about EPI formation. The procedures they have a little difference.
Oh, I see.
Yeah.
I see. Questions? Anyone?
Okay, maybe I'll start with another question. Professor Ji, great talk. So have you ever thought about, for this in vitro culture system, right?
Yes.
If you change the culture media or condition, would they behave differently?
Yeah,
and how similar is this in vitro system when compared to
Yes, mostly is similar to human culture system, but you have to do some little changes to plus some chemicals, maybe much better. So far, we still improve this culture system. We hopefully not just culture the monkey embryos to 20 days. We hopefully can culture to more days, maybe 25 days, 30 days. And so we can clearly answer the questions about the gastrulation for primates.
Any more questions from the audience? We do have quite a bit of time, so or perhaps if anyone has any questions for any of the speakers. Okay. Well, if there are no more questions, we thank Professor Ji for giving a fantastic talk, and I guess we can finish early. Feel free to hang around if you have more questions. If not, thank you. Thanks for attending the session.
Thank you.
Thank you.